My title is Mass Spec of Glycomics of Tissues and Biofluids, and I put the antibody on the title slide. And as mentioned by Mike, I'm here at the LUMC in Leiden at the medical center, and I'm heading the Center for Proteomics and Metabolomics. And my first few slides are on introducing the center. So the medical center here, the LUMC, has chosen for having one central mass spec omics research unit, which is our CPM. And we are developing technology wherever needed, and we use high-end mass spec, NMR, and in general analytical technology for answering basic life science questions, but also for finding markers which are suitable for clinical translation. Our center is organized in a couple of groups or a bunch of groups. So we have a mass spec imaging group. We work on small molecules, metabolites, and lipids. We work on proteins, bottom-up proteomics for a big part. We have my glycomics team, and we have bioinformatics support, which is getting ever more important. Now, today I will try to focus a bit on data and data structure. So what type of data are we generating? But all of that is driven for a big part on the questions we try to answer. And those are related to structures of glycans and glycoconjugates, but also to their biological function. And the mass spec is our center stage technology. And I'll start with looking at isomers of glycans, differentiating those isomers, and doing that for specific cell types. I think this is very important that we know what we're looking at. So this can be a specific protein, but certainly also we need this cell type specificity because human cells do very different jobs in terms of glycosylation. And for that, we look at specific tissue regions which are dominated by one cell type. So we did this for colorectal cancer, and this is just on the workflow we are using for answering these questions. So we have sections of tissues, and here you see the epithelial layer of a colon, which we would then cut out by laser capture microdissection and analyze its glycome. We do that the very same for a tumor. So we cut out the cancer cells, which are related to these normal epithelial cells, but are then degenerated indeed. And we take these LCM prepared tissues, we homogenize, we immobilize on PVDF membranes, we cut off the N-Glycans, we chemically release the O-Glycans, and we analyze all these glycans by LCMS and use PGC, porous graphitized carbon, for resolving isomers, which is a key aspect of this workflow. And we need quite some cells here, 20,000, but you can prepare those in a tedious but feasible manner from these tissue sections. Now, two aspects I want to highlight with this slide here. So one is that the areas are different. So we have here the cancer cells, colorectal cancer cells prepared from patient tumors with this LCM method. And we have the stroma, so kind of the fibroblasts surrounding it. And we see very distinct glycosylation profiles for these two different tissue areas. And we can measure these glycomic signatures for the N-Glycans, but also for the I will try to use a laser pointer, but also for the O-Glycans as shown at the right-hand side in a repeatable manner. And that's important, of course. And we see that indeed the glycomic signatures are very different for these different areas, which are maybe just 100 micrometers apart, but dictated by very different glycosyltransferases and glycomic signatures. So here we're now comparing the carcinoma, so the cancerous area with the normal colon of the very same patient, just a few centimeters apart. And what you see here is that we have core 2 O-Glycans with Lewis motifs, but also Lewis motifs. 4 of them are highlighted here, and these we only find in the cancer, but we do not find them in the normal colon. And this was astonishing for us because we expected some minor changes, maybe some up and down regulation, but this was more like a black and white picture, which we obtained here, at least for this patient. But of course, we wanted to look more broadly. So we did a number of patients, and we did then this PCA analysis, unsupervised data analysis. And there we see the normal mucosa samples, all of them clustering. And then the cancerous samples were also clustering here in blue. We looked at the stroma, so the supportive tissue surrounding the cancer. We looked at immune cells, and we were surprised to have quite distinct glycosylation profiles as reflected in these score plots. If we look in more detail, so which type of O-glycans were we detecting, because this is now really only focusing on O-glycans, this part of my presentation. So we saw 172 different O-glycans, of course, not all of them in all tissues and in all samples, but we found 100 O-glycans only, 100 O-glycans only in the cancer. We didn't find them in the normal tissue and also not in the stroma tissue. So these 100 O-glycans, how do they look like? They are, for big part, they are called 2 O-glycans, as I showed for the adenocarcinoma, and they have, if we look here, terminal 2,3 salivation, we have quite a bit of Lewis type structures in there. So quite a number of O-glycans only in the cancer tissue, not in the healthy surrounding tissue epithelium. And if we try to summarize this now, then this might be a way of grasping the data. So we see that indeed quite some of these O-glycans are not present in normal mucosa. They are present in the cancer, and intriguingly, they are more prevalent in invasive cancer tissues as compared to non-invasive cancer tissues. So this suggests that this may even associate a bit with disease severity. So we have indeed these core 2 glycans with the terminal Lewis and Sally-Lewis motifs. And another way of summarizing the very same data is then these box plots here. So we see the core 2 enhanced in cancer, or basically, it's almost black and white, so it's in cancer, but hardly in normal tissue. We see the 2,3 salivation enhanced and the combination thereof. And then on the other hand, core 3 glycans are high in normal tissue, but very low in cancerous tissue. And we try to come up with a biosynthetic pathway in line with the textbook knowledge. And we also used some transcriptomics data, public data, and pasted them on top. So what we see is indeed the core 2 pathway here with the terminals Lewis and Sally-Lewis motifs. We see the truncated O-glycans up in cancer, but we see indeed core 3 mainly in the normal mucosa and way lower than in the cancer. So, what is the way forward now with this? We were encouraged to develop therapeutic antibodies. Hopefully, this will help then to fight maybe advanced cases of colorectal cancer, maybe even metastases. So we're also looking whether metastases have a similar glycomic signature. Okay, so this is all I wanted to show on colorectal cancer and tissue-specific glycosylation. Now the next part is really more on another level of specificity, and that is which protein are we looking at and which site are we looking at. Glycans do very different jobs depending on where they are located. And I will actually look into antibodies and not only look at their structure, but also show data on binding, which explain immune activation. So if we look at the IgG molecule, we know that in the CH2 domain, we have a conserved N-Glycosylation site, which can carry various different N-glycans, mainly diantennary complex type glycans with a core-fucose. And these glycans matter for a lot of functional aspects of the antibodies, and that's shown down here. So here we look at the binding of the antibodies to the 3A receptor, simple binding assay, microtubule-based. And so what we see here is if we express the antibodies, and we did this together with our collaborators at Sanquin, Gesto Vidason and colleagues. So if we express these antibodies in 20 different glycovariants, the variants with a core-fucosan do hardly bind to the 3A receptor, these ones here, but the variants lacking the core-fucose are quite good binders to this immune receptor. And then we see that a gradient of galactosylation on top matters. So the more galactose is present, the better these antibodies are in binding to the 3A receptor. So this is a binding assay. If we now look at an in vitro cellular assay with the cell activation and cytotoxicity, then we see that also immune cells are more potent in cancer cell killing, depending on the antibodies you offer, which bind them to the cancer cell and recruit the immune cells activated. So this is really a simple glycosylation switch, which determines cytotoxicity and immune activation via the fucosylation. Now, my lab is doing putting quite some efforts into establishing assays, new assays. And what we did here is we said, of course, we can try and produce all these glycovariants one by one and then assay them one by one. But what about having a multiplexed assay? And what we did is actually we put the 3A receptor on a column. We flushed the complex mixture of glycoforms across the column. We then look at the retention, so higher retention reflecting more higher affinity binding. And we use a mass spec for detecting all these intact antibodies flushed across the column. And what we then see is very much in line with what the glycoengineering told us in the previous slide. So we see that indeed all those variants which are highly fucosylated, all those glycoforms have a low binding, whilst the variants having, at least on one arm, a lack of fucose show a high binding, like this G0F G1 glycoform, which is schematically shown here, is here in this ladder eluding peak. And then we see that on top galactosylation matters. So here, lack of galactose comes with a very early elution, very low binding, and the variants having higher galactosylation show at least slightly increased binding. So galactosylation, but most severely fucosylation or afucosylation matter for the 3A receptor binding. So this was on the in vitro functional assays. And I'll now move into rheumatoid arthritis and show a bit of experiments we did there on characterization of the pathogenic autoantibodies. So these are antibodies against citrulline, and these antibodies are in part produced by B cells, which are close to the inflamed joint. So these antibodies are then secreted into the synovial fluid. The antibodies are prevalent, so most of the RA patients do have these autoantibodies. And they are directed against citrulline, so an enzymatic modification of a peptide backbone could be any protein in this inflamed joint, which is modified by the enzyme, and then the epitope is generated and targeted by these antibodies. Now, we wondered how heterogeneous are these antibodies? What is their glycosylation? Could this influence their pathogenicity? And so we really established a number of assays to look at antibody structure and glycosylation. And in this case, we focused mainly on the FC portion. So for that, we cleaved the hinge region with IDAS, with this enzyme, and Konstantin Blöchel, who is on the call, was leading this, or mainly performing these assays. So we got a very valuable patient cohort of 11 rheumatoid arthritis patients, for which we had not only plasma, but also fluid from the inflamed joint, so synovial fluid. So we could look at the site of inflammation, what is the antibody repertoire there. And now to show you some data on this, we did this for the autoantibodies, also for bulk antibodies. We generated these FC half portions, CH2 and CH3 domain with a glycanon, and we did an LCMS analysis to assess glycosylation. And we did this in this case for one patient. I show the data where we have the plasma IgG, total IgG, and then also the autoantibodies from plasma. And you see that here the subclasses are differently involved. This is the overall subclass profile of this patient, and this is the autoantibodies. And for the autoantibodies, we see that it's mainly the IgG1 subclass contributing. And this is quite similar for the synovial fluid. Also there we find the autoantibodies. So they are in the synovial fluid, but also in the circulation. And again, it's mainly the IgG1 subclass contributing. We then looked at another patient, and now this patient is heterozygous for all the heavy chains. So it has two heavy chain variants on the two chromosome sets encoded. And here we can look now at, in light blue, at the IgG1 allotypes, and we can do so for the bulk IgG, but also for the autoantibodies, and we see a difference in these peak ratios. So you see that this O3 version is becoming more dominant for the autoantibody in plasma. It also becomes more dominant for the autoantibody in the synovial fluid. So we see a differential allotype allotype usage of these antibodies for the autoantibody versus the total IgG. You can do this now for a number of patients, and this is consistent. So the allotype usage is different for the autoantibody versus the total IgG. And now we move to glycosylation. So just to show you that we can look at different attributes of proteins. So we can look at these allotypes, but now also at glycosylation. And if we do a non-supervised analysis of all the glycosylation information of these alloantibodies and total IgGs, we see that total IgGs are quite different from the pathogenic autoantibodies in terms of glycosylation. And then we also see that the autoantibodies dependent on the biofluid we're looking at are also different. So synovial fluid is different from plasma autoantibodies, ACPA antibodies. And this is also reflected here in the loading plot, but I think it's maybe easier to just look at these simple graphs here. So the first objective is to look at total versus autoantibody, and that's shown here. So we see a difference in fucosylation. So here we have the total, here we have the ACPA, the anti-CCP autoantibody, and for the total we have a noticeable amount of non-fucosylated antibodies, but these are virtually absent for the autoantibody. So the autoantibody is almost fully fucosylated, whilst here we have some lack of fucose for the total IgG. And that's quantified down here. So afucosylation is indeed strikingly different for the autoantibody versus the total. And this is not only the case for plasma, but also for synovial fluid. We also have differences in bisection and in site occupancy. So overall, these sites are very highly occupied. The CH2 domain glycosylation is very prevalent, but we see that still there's a difference in site occupancy for the autoantibody versus the total or bulk IgG. And again, this is conserved for plasma and for synovial fluid. Okay, and now another angle of comparison is the comparison of the autoantibody for the two different biofluids. So we can compare the plasma with the synovial fluid. And that's shown here. And also here we see quite some striking differences. So the lack of glycosylation, the A-galacto variants are more prevalent for synovial fluid ACPA as compared to plasma ACPA. And also for silylation, we see accordingly these differences. Now, for IgG3, we do not only have the CH2 domain glycosylation, but also a CH3 domain glycosylation. And that's shown here. So we have two different domains, both of them glycosylated. Here, the CH2 domain glycosylation, which you find on virtually all subclasses, but then on IgG3, as I said, also CH3 domain. And then we have an integrated pattern of these two glycosylation sites given here. And here Konstantin is just demonstrating that we can measure this with very high precision. But if we now look in the next slide at different patients, then we see that, again, the autoantibody is different from total IgG in terms of this CH3 domain glycosylation. And the difference lies, for example, in hymenos glycans, in their relative abundance. So they are more abundant for the autoantibodies versus total IgG. But also site occupancy is different. So plasma cells producing antibodies obviously do this differently for the for bulk IgG as versus these autoantibodies. Yeah, so this is on the autoantibodies. We are studying these in way more detail. We also look at Fab glycosylation, which I'm not showing here. But what I hope I could demonstrate here is that we can use intact mass analysis, separation techniques, and reveal a lot of features. We can look at proteoforms, different glycoforms, different glycosylation sites, and get a comprehensive overview on the molecular makeup of these antibodies, and in this case, pathogenic autoantibodies. Okay, now in the third part of my talk, I want to look at higher throughput analyses. And in this case, we are dealing with hundreds of samples, of clinical samples. And I chose, again, antibody profiling. And so what we did for many years is look at specific antibodies. Here we did not do intact antibody analysis, but glycopeptide-based analysis. And what we focused on for a big part is the level of fucosylation. And I showed you this is functionally important. So we saw for a bunch of clinical conditions, I will not go into detail here, but also for quite some viral infections, pronounced afucosylation for the antibodies. This is CMV, but also for HIV, we saw that quite some patients have low levels of fucosylation on these antiviral antibodies. But for other viral diseases, we missed this low fucosylation signature, like for parvovirus. We also saw for hepatitis B infection, for example, that the natural infection comes with a diversity of fucosylation. So some patients low fucosylation, whilst the vaccination, which is a soluble vaccine domain, protein domain, comes with a high level of fucosylation. So initially we were puzzled by this, but we came up with a hypothesis, which until now holds true. So soluble antigens and capsid viruses induce high level of fucosylation, whilst membrane-associated antigens like for SARS-CoV-2, but also for HIV and CMV, here we have antigens in cellular membranes, in viral membranes. So these membrane antigens induce low levels of fucosylation, or at least they can do so. And then indeed, this translates into different immunological activities. And just two recent findings, we also found that in transplantation and in transfusion, we also can have these low fucose signatures, and those can lead to immune activation and, for example, kidney damage. So a recent collaborative paper we published. But I'll now focus for these low fucose responses on COVID-19. So what we did here is we profiled the antibodies against the RBD domain or anti-S antibodies. And here we saw for one patient that an early time point was quite different from a late time point. So here, for example, we have a glycoform lacking fucose at the early time point, while this glycoform with a lack of fucose is down at a later time point. So we have an initial low fucose response, which normalizes within some days or weeks. And this is now only a single patient, and the data I'm showing here are generated by affinity purification of the antibodies, tryptic cleavage, and then measuring of the glycopeptides. But then we went in a certain direction by showing with in vitro assays that this fucosylation indeed matters. So the fucosylation assay, the in vitro assays use these antibodies, in this case, they're monoclonal antibodies against the S protein. And the antibodies are applied with high and low fucosylation levels. And we see that indeed the low fucose variants, which are here and here, are the ones which are most potent in activating immune cells and in inducing inflammatory cytokines like IL-6. So the in vitro assays substantiate the idea that this low fucosylation may contribute to inflammation in the COVID setting. And we then, triggered by the data on early versus late responses, we then looked at the whole time course for 150 patients roughly. So we followed all these patients as shown here, and we saw indeed that the early time points when these patients came into the clinics with massive symptoms, these early time points for many patients showed low fucosylation, and then in the course of the infection, the fucosylation levels were normalizing. We also saw that early time points came with skewed galactosylation, so high galactose, high silylation, low bisection. So we had a very skewed early glycosylation profile. And that's summarized here with a more focus on the early time points. So indeed, the anti-S earliest time points, so the initial antibodies have this low fucosylation. We then looked at clinical associations. Now, strikingly, the fucosylation did not really show us a good association. So here we only simply stratified for ICU admittance, so which patients eventually got into the intensive care unit and which didn't. And we did not see a clear difference in terms of fucosylation. We did see so at the moment of hospitalization that the level of glycosylation, of bisection, and of silylation in part predicted whether people would get into an ICU. And interestingly, the more skewed the glycoprofile, so the higher the glycosylation, the higher the silylation, and the lower the bisection, the more skewed the profiles, the more people were protected from ICU admittance. Now, from infection, we then moved into vaccination and looked whether the glycoprofiles of induced by these mRNA vaccines would matter. And what we found is indeed also for vaccination, we have an initial low fucose response, which then in the course of the response and with a booster, indicated here in gray, normalizes. So we looked at bisection, glycosylation, silylation. Virtually, we saw that the mRNA vaccination very much mimics, in terms of the induced glycoprofiles, the infection. And now we then looked at associations with the titers, and that's shown over here. So the lower the initial fucosylation of your immune response, the higher the titers are. It's not a very strong association, but it's a clear association. So the lower the fucosylation, the higher the titers will be after the booster vaccination. So for achieving high titers, the initial low fucose response seems to be beneficial and seems to matter. So just to summarize the part on these viral infections, and particularly COVID-19. So we think that this phenomenon that we can have these low fucose responses really matters, and that these low fucose responses can indeed translate into a more profound inflammation, higher antibody titers also in the end. So they can be in a way beneficial in mounting a strong immune response, but they can also lead to massive tissue damage. And we imagine that part of the lung tissue damage and the organ failure could be induced by these low fucose responses in the case of COVID-19. But in a way, we think this is a double-edged sword. So these low fucose responses induced by membranous viruses can be helpful in clearing the infection, but can also lead to excessive inflammation. Now, in the last part of my talk, I want to look into even higher numbers of samples, clinical samples. And now I'll go for an approach where we indeed ignore the protein part and simply look at released glycans. And for this approach, we choose blood, and we look at blood proteins in the form of plasma or serum. And I will now introduce the approach. So we release the N-Glycans from serum or plasma. We derivatize salic acids. So this is something we do now for 10 years already in our lab. So we induce a lactone for the 2,3-linked salic acid, and we induce ethylation for the 2,6-linked salic acids on these N-Glycans. We do a helix cleanup, and we do a MALDI experiment to measure these plasma, serum, and glycomic signatures. And here is a typical spectrum we will acquire by MALDI mass spectrometry. We will measure sodiated glycans. And what you see now is the benefit of this salic acid derivatization. For example, here we have a cluster of 3 peaks, and they differ in the linkage of the salic acid. So here we have 3 times the 2,3-linked salic acid indicated by the left tilt. And then we have another variant here, which only has 2 2,3-linked salic acids. And here only one of the salic acids is 2,3-linked, while the other 3 ones are 2,6-linked on this tetraantennary glycan. And we can do this differentiation of salic acid linkages all over, also for the diantennary glycans. So we get a differentiation of these linkages, which are functionally important. And I now chose a very recent application where we looked into fatty liver disease, non-alcoholic fatty liver disease. It's a highly prevalent disease with the prevalence vastly increasing in the last decades. as indicated by the red brownish colors here on the world map. Now, there's multiple factors which contribute to this non-alcoholic fatty liver disease, or NAFLD. And if you look at the disease course, so you start with a normal liver, and then in the end, your liver gets fatty, you get fibrosis, you get cirrhosis, you can end up with hepatocellular carcinoma. So this is a very serious condition. And what the patients we were looking at were stratified according to the level of fibrosis. So they were ranging from F0, virtually no fibrosis, to F4, very advanced fibrosis. And this is reflected then also in, I mean, you can translate this into a time axis of disease progression. Now, our clinical collaborators, they asked us for differentiation between patients, and they were particularly interested in picking up these fibrosis patients, F1 to F4, and differentiating them from other fatty liver disease patients. And we looked at glycosylation traits as indicated here. So we looked at antinarity of complex type glycans at 2,3 versus 2,6 cyanylation, levels of glycosylation, and ternary fucose bisection, and so on. And this is just a summary of the data which we obtained. So strikingly, it was the salic acid linkage which made the difference. So here the ethylation is reflecting 2,6 cyanylation, and that one was increased in the disease, whilst the 2,3 linked salic acid indicated by lactonization and the big L was down in the disease. Now, here, just a different way of looking at this, and we were very happy that we could replicate these markers. So we had a discovery cohort, a replication cohort, and we had a perfect replication of these N-glycomic markers. And then we looked into this differentiation of the fibrosis stages from non-fibrotic cases, and that's shown here. And we found, for example, that this single feature here, so the 2,3 cyanylation of the tetra and ternary glycans, as indicated here, is sufficient to get a very decent differentiation of the fibrotic from the non-fibrotic cases. Now, looking a bit more broadly, we not only looked at glycomic signatures of serum plasma for NASH and NAFLD, but we also looked at other diseases like colorectal cancer, inflammatory bowel disease, and rheumatoid arthritis. We did quite some attempts of summarizing these data. And here you have the glycomic profile of serum plasma. We summarized literature to see which proteins are likely contributing to these glycans and which diseases show a up or down regulation of these glycans. And we don't only have 3 or 4 diseases now that we covered, but a range of different conditions. And we now, for the coming year, try to put effort into integrating all this information and get a kind of global disease map, glycomic disease map, where we see with which inflammatory triggers and which disease-associated changes we would also get glycomic changes. So kind of a glycomic disease map. Now, what is interesting is that we can measure these plasma glycomic signatures, of course, from serum plasma, from processed blood, let's say. You can also simply use dried blood spots and get a virtually identical glycomic signature, and that's shown here. And the nice thing about these signatures from dried blood spots is that you can store these dried blood spots for weeks at room temperature, and you don't see a clear change of the glycomic signature. So these are very robust and stable, and give quite a potential, for example, sampling at home, shipping these dried blood spots, and then maybe measuring at a omics lab and getting clinically relevant information. So this brings me to the end of my presentation. I showed quite some technology which we developed and we used for elucidating glycan and glycoconjugate structure, but also for looking at function. I also want to look ahead a bit. So next to mass spec, I'm a mass spec believer. I think that also mass spec imaging, spatially resolved ion mobility, different spectroscopic methods, but also new bioinformatics solutions will be key for further advancing the field here. And in terms of function, we see that integrating different omics, also using microarray data, and then going to the lab and doing some functional assays, knocking out some genes, some molecular biology, also using chemical biology tools. So these are very fruitful approaches to get more functional insights into glycoconjugates. Now, a few conclusions. So mass spec is powerful for glycomics, both structural and functional glycomics. And what I like about the intact or native glycoprotein analysis is that it's comprehensive. So you get an insight into different proteoforms, glycoforms, how they interact in changing protein structure and function. What astonished us was finding that antibody glycosylation in infection and vaccination is quite dynamic. So we saw changes within a few days, whilst if you merely look at antibody half-life of a couple of weeks, you would expect more slow, less of a dynamic pattern of antibody glycosylation. And this is, of course, very relevant because antibody glycosylation does define functionality. Now, for tumor glycosylation, we need the cell type resolution because a cancer cell is different from a fibrotic, fibroblast which surrounds it. So you need this resolution by laser capture microdissection. And then we were astonished to see very highly skewed O-glycosylation signatures with a therapeutic potential in CRC. And then for many, many diseases, we see that the blood glycome is highly skewed, shows quite some differences. And we think that for liver conditions, but also for other conditions, this may have diagnostic and prognostic potential. And of course, we would love to get more insights into the mechanisms, underlying mechanisms. Okay, this brings me to the end of my presentation. I want to acknowledge the funders, but most importantly, my great team. So many, many team members were involved. Katharina, Noache, Konstantin, Elena, Christoph, Thomas, Stefan, David, Guinevere, and so on. So a long list of lab members, and I really appreciate all of your contributions. Then our in-house collaborators on rheumatoid arthritis, but also on the hepatology. Gestor with his team at Sanquin, our industrial collaborators, Dietmar and Markus. And then with this, I want to thank you for your attention.